Hello, in this video I'm going to be talking about blood group and genotype. Unlike my previous videos, this particular video is not specifically meant for medical students. Rather, it's meant for a group called Young Wives Association. So if you're a medical personnel or medical student, you might not really want to bother yourself with this video because it already contains all the informations that you already possess. So what is blood group? Blood group is a classification of blood that is based on the presence or absence of some particular molecules or substances or whatever you might want to call them that are present on the surface of red blood cells of individuals. But medically, these substances or molecules that are used to classify blood into different groups are called antigens. And these antigens, we inherit them from our parents, one from each parent. Now, the question will arise, is it only humans that have blood groups? No, it is not only humans that have blood groups. All animals that have blood also possess blood groups. And their groups, however, are different from that of human beings. For example, in cats, you have at least 3 major blood groups. The classification, of course, is different from the way we classify blood group for humans. Then for dogs, dogs have at least 4 blood groups. And when you come to cows, cows have about 800 or more blood groups. But because of differences, human beings, animals, human beings, dogs, cows, cats, they belong to different species. So you cannot transfuse humans with animal blood. Likewise, animals cannot receive human blood. So how do we arrive at these blood groups? They are determined by the presence or absence of these molecules that are called antigens. And based on these antigens, two main systems are used to identify human blood groups. And these two systems are designated as the ABO system and the rhesus or RH system. So you have the ABO system separately, you have the rhesus system separately. However, these two groups are always combined when describing an individual's blood group. If you are using the ABO system alone, we have 4 different blood groups possible. And these four different blood groups are the A, B, AB, and O groups. Then if you are using the rhesus system, 2 blood groups are possible. It's either the blood is rhesus negative or it is rhesus positive. However, we combine the two systems to arrive at human blood group. So in combining the two systems, we now have A plus, B plus, AB plus, O plus, O negative, AB negative, B negative, and A negative. So what that means is that we have 8 blood groups in humans. So human beings can belong to any of the 8 blood groups that we just mentioned here. Can blood group be used to determine parentage? In some cases, yes, it can. You have instances where people will come and say, they say, I'm the father of this baby, but I'm not the father. But I want you to take my blood, check my blood, and check the blood, to check the baby's blood to confirm if I'm really the father. One thing we must mention is that blood group cannot be used to confirm paternity, but in some cases, it can be used to exclude a man as the father of the baby. For example, you have a man that has blood group AB, and then you have a child that has type O blood. Definitely, that man cannot be the father of that child, because if he's the father, you will either find the A or the B inside the child's blood. But this child is having O, it doesn't have A, it doesn't have B. And the father must pass either the A or the B to his child. So in this case, with the child having blood group O, definitely the man that has blood group AB is not the father of that child. So the absence of either the A or B in that child automatically means that that man cannot be the father of that child. So what are the clinical significances of blood groups? Number one, blood group is important to know an individual's blood group in cases where the person receive blood, where the person require blood transfusion. What does that mean? If someone has anemia, that is the level of blood in the person is low, or the person has an injury and lose blood, or in some other situations where someone require blood transfusion, you need to know the blood group of that individual. Then you need to check the blood group of the person that is going to donate the blood to be sure that it matches the blood group of the person that is going to receive the blood. The person that is donating the blood is called donor, and the person you are going to transfuse the blood into is called recipient. So you have to be sure that both the donor blood and the recipient blood match. If that is not the case, if the two blood groups do not match, the recipient is going to have some very serious blood transfusion reactions that might even lead to the death of that person. So, for blood transfusion cases, blood group is very, very important. Then, in pregnancy, it's also important. Blood group is also important in pregnancy. There are some women that are rhesus negative. And the problem with rhesus negative mothers is that if they marry a man that is rhesus positive, it is possible that the child, the baby inside the womb could be rhesus positive also. And if that baby in the womb, if his baby is rhesus positive, then the mother will develop, the immune system of the mother will develop some substances or molecules that are called antibodies. And these antibodies will now destroy the blood of the baby that's inside the womb. And this can lead to serious complications. It can lead to anemia of the baby inside the womb. It can lead to premature delivery of the baby. It can even lead to the death of the baby. So it is a very serious condition. However, it is very possible to detect it even before conception or during pregnancy also. It's possible to detect it. And when it is detected, there are things that can be done to prevent the mother from developing antibodies that will target the blood of the baby inside the womb. We call it rhesus isoimmunization. So it can occur after delivery, or no, It can occur during delivery. It can occur following a miscarriage. It can occur following an ectopic pregnancy, or it can occur following an abortion. So this is why it is important for women to know their blood group and also to know the blood group of their partners so that they don't develop rhesus isoimmunization. And when you check, maybe after delivery, you check the woman's blood group, the woman is rhesus negative, and you check the blood group of the baby. The baby is rhesus positive. Or you check the blood group of the woman, she's rhesus negative, and you check the blood group of the husband. The husband is rhesus positive. After delivery you should check the blood group of the baby. And if the baby is positive, is rhesus positive, then you have to start the mother on an injection. You have to give that injection within 72 hours of delivery or of miscarriage or of abortion to prevent, to neutralize whatever antibody the mother must have formed so that she, she will not have, her babies in subsequent pregnancies will not develop complications or problems arising from rhesus isoimmunization. So now let's look at the different blood groups that are available and the different groups that can donate blood for them. For O negative individuals, they can only receive blood from O negative donors. Okay, then O positive individuals can receive transfusion or can receive blood from O negative or O positive individuals. Individuals who are A negative can receive blood from O negative and A negative individuals. Then people who are A positive can receive blood from O negative, O positive, A negative, and A positive people. Then those who are B negative can receive blood from O negative and B negative individuals. Then those who are B positive can receive blood from O negative, O positive, B negative, B positive. Then those who are AB negative can receive blood from O negative, A negative, B negative, and AB negative individuals. And those who are AB positive can receive blood from O negative, O positive, A negative, A positive, B negative, B positive, AB negative, and AB positive. So if you go through that, you see that those who are negative can only receive negative blood. Look at O negative, look at A negative, look at B negative, look at AB negative. You see that they can only receive blood from individuals who are also negative. Otherwise, if you transfuse them with positive blood, they will have serious transfusion reactions, which I must emphasize again, can lead to their deaths. So individuals who are O negative are called universal donors because they have no antigens that can trigger a reaction in the recipient. Because of that, they can they can donate blood to anybody, or their blood can be transfused to any blood group at all. Then individuals who are AB positive are called universal recipients because they have no antibodies to either A or B or negative in their blood. So they can receive blood from any blood group. Quickly, let's look at genotype. Genotype is the genetic makeup of the individual, and it describes the type of proteins that are found in red blood cells. And in humans, 6 main types of genotypes are described, and these are AA, AC, AS, CC, SC, and SS. People with SS and SC genotypes are prone to having sickle cell diseases. And just like blood groups, genotypes are also inherited from parents. One gene is inherited from each parent. So if you have genotype of AA and your partner has genotype of AS, you can give birth to AA or AS, since the child will inherit one gene from you and another one from your partner. Now let's look at this diagram for you to understand that properly. Now look at husband. Husband is AS, wife is AS. So if they conceive, if the woman conceives, the baby will receive one from the husband and one from the wife. So if the baby receives A from the husband and A from the wife, then the baby will be AA. On the other hand, if the baby receives A from the husband and S from the wife, then it will be SS. But if the baby receives S from the husband and S from the wife, then the baby will be SS. So if you look at that diagram properly, for each pregnancy, there is a 1 in 4 or 25% chance that the baby will be AA. Then there is also a 25% chance that the baby will be SS. And there is a 50% chance that the baby will be AS. This risk is only for a particular pregnancy. But understand that I'm not saying that if the couple have 4 children, that maybe only one will be SS and the others will be either A or AS. No, this risk is described for each pregnancy. Okay, let's understand that very, very well. So does genotype change? No, genotype does not change. The only way genotype can be changed is by bone marrow transplantation. But that is a subject for another discussion. There are tests that can be done while the baby has not been delivered to determine the genotype of the baby right inside the uterus. And these tests are done for couples who are carriers of the S trait or who have had a previous baby who is SS. So these tests are done early in pregnancy. The first one is called chorionic villi sampling. And for chorionic villi sampling, it is done between 11 and 14 weeks of pregnancy. Then amniocentesis, this is done between the 15th and 28th weeks of pregnancy. And there's a third one called pre-implantation genetic diagnosis, which is used in IVF. However, these tests are very, very expensive. As at last year, I know that the chorionic villi sampling and amniocentesis goes for over 800,000 naira. I don't know how much it costs currently. Now, there are some questions that arise from this lecture. Number one, what are the symptoms or signs of SS in childhood? Okay, can you know whether your baby is SS right from childhood? Well, a baby that is SS is likely to be having frequent fever, is likely to be having frequent infections, especially lung infections. Then there could be swelling of the hands and feet. Then there could be episodes of pain, body pain, bone pain, then anemia. Okay, anemia will be present. Then the growth might be delayed, and some baby might have vision problems. So these are some of the signs that you might notice or that might make you suspect that a baby or a child is having is SS. Then another thing, some people have asked that is it true that people who are SS are immune from malaria? Yes, it's true, because the sickle cells offer some protection against malaria, but this protection is not absolute. And people who have SS are also at risk of developing some very severe forms of malaria. Another question that have been asked is whether there is any special food for SS patients to eat to prolong their lives. No, there's no special food for SS individuals to eat. However, they need to be going to hospital regularly for checkup, okay? And some might be placed, they will be on antibiotics to prevent development of infection. Then vaccinations also, the vaccinations have to be taken seriously, okay? One of the complications they have, especially in childhood, is pneumonia, okay? So there are vaccinations that are given to reduce the risk of developing that type of pneumonia in individuals who are SS. Then there are antibiotics that are given to also reduce the risk of developing infection. So no special food, but of course, the food must be balanced, just like you who is not SS, just like your own food too. Your own diet should be balanced diet also. So uh, regular visit to the hospital for checkup is mandatory. And when these are done, when the child or the individual is under regular monitoring and under regular supervision at the hospital, then he can live an active life and can live almost as long as individuals who do not have the SS genotype. Is there any other question? Yes, there's another question here. Are there different types of AA genotype? No, there are no different types of AA genotype. AA is AA. There are no subtypes. There's no AA1 or AA2 or AA3. AA is AA. So these are some of the things we'll discuss here for this lecture. I don't want to continue so that the video would not be too long and boring. Okay, thank you very much. Thanks for watching.